Hello and welcome to the 25th episode of the podcast. Today I'm going to be opening up a mini-series on the evolutionary history of life. The first episode of this history, naturally, is all about the first life forms, the first cells, and the earliest kinds of communities and food chains that they created. To understand how the first forms of life emerged, we must understand the environment this life was born into. We must understand what minerals in the crust and what gases in the atmosphere our earliest ancestors were exposed to. The first era of the Earth's life is called the Precambrian, and it's a massive expanse of time that began with the formation of the Earth 4.5 billion years ago, and it lasted up until the Cambrian explosion about 540 million years ago. Thus, the whole Precambrian lasted nearly 4 billion years. Even though this eon constitutes nearly 89% of the planet's lifetime, we know barely anything about it. It took about 500 million years for the first life to emerge. And after that, for the next billion years, what little life there was remained as single cells, but it gradually formed colonies, or just giant masses of single-celled critters all lumped together. For the next 1.5 billion years, these primitive cells would evolve greater sophistication in their organelles and their DNA. They would evolve symbiotic relationships with the organisms around them, and they would also evolve multicellularity. Near the end of the Precambrian, all the evolutionary steps building momentum over the last 2 billion years would create the first multicellular life, the first plants, the first fungus, and the first animals. None of these life forms would start producing fossils en masse until the very end of the Precambrian. Fossils of some of the earliest cells were rare to form, but some have been recovered and studied extensively. Cell fossils are really hard to find, and small multicellular organisms aren't much easier. Furthermore, most multicellular organisms that emerged during the Precambrian were soft-bodied. They had soft tissues and nothing at all like bones or exoskeletons, and so they were highly unlikely to become fossilized in the first place. With that said, some of this might sound familiar to episode 2, where I introduced the basics of biochemistry and the chemical environment of life on a molecular level. The story of life is a story of all living things, including us. We have all descended through millions of generations, across billions of years, from the first cells capable of self-reproduction. When I say we, I mean myself, you, our families, our friends, their families, their friends, and the rest of humanity. I mean the animals, bacteria, and archaea, and everything else that crawls, or squirms, or flies, or swims through the thin band of life clinging to the Earth's surface. The story of all of this life begins 4.1 billion years ago, when the first groupings of biomolecules organized into the first cellular membranes to maintain a stable, self-reproducing entity. These first cells would persist through time. Their descendants would evolve more complex traits like eyes and fins and central nervous systems. This life would crawl from its ocean womb onto dry land, facing a whole new world with new threats and unknown dangers. The Precambrian era is broken up into several periods, and the first period is called the Hadean, and it began with the fiery birth of our planet nearly 4.6 billion years ago. The Hadean was a time that lasted for about 6 million centuries, and it was filled with earthquakes and volcanic eruptions and perpetual asteroid impacts, but these tapered to a gradual end about 4 billion years ago. This isn't to say there was some hard delineating event 4 billion years ago, just that it's the time frame created by geologists based on the evidence from their field of study. During the Hadean period, lava flows perpetually repaved and resurfaced the Earth. These fields of flowing and cooling lava were propagated by the heavy volcanic activity of the unstable and still developing tectonic plates. But the surface of the Earth wasn't the only source of chaos. The early solar system was characterized by rings of debris, of fields of dust and rock. Earth began as a small mass, which grew by accumulating the matter that happened to be in its orbit. The Earth grew quickly, as matter wasn't burned up in the atmosphere. There wasn't an atmosphere, at least not yet. The planet grew, and its gravity swelled in size, gulping down all the matter it could reach. In time, the Earth had cleared its orbit, consuming virtually all of the available dust and rock. As the Earth grew, the interior pressure increased until it was a ball of liquid iron, with a primitive mantle and newly formed tectonic plates. These plates were unstable, and they broke often, spilling magma onto the surface of the Earth, like the blood of the planet seeping out from massive cracks and tears. From the surface of the Earth looking up, this period of dust accumulation and orbit clearing would have been characterized by extremely heavy asteroid bombardments. The sky would have been carved up by streaks of black smoke and fire that smashed into the surface throughout the day and night. To make this extreme image even more intense, the formation of the moon involved a glancing impact of literally planetary proportions. At some point, a planetary body known as Theia collided with the Earth. Theia is predicted to be roughly the size of Mars, so the impact would have been incredible. The force of the impact outright vaporized a huge portion of the Earth's crust, throwing an immense layer of vaporized rock into the atmosphere, and this created a debris field in space. The debris field contained about 20% of Theia's mass. Some of it would eventually fall back to Earth, while some of it would just fly off, lost into space. But about 50% of this debris would eventually coalesce at a stable point in orbit around the Earth to form Luna, the moon. The layer of rock vapor in the atmosphere settled within a couple thousand years, while the heavier material in the molten crust sank deeper into the mantle. The Earth's atmosphere at this point in time is a hellish 446 degrees Fahrenheit. It's choked with carbon dioxide and nitrogen. As for oxygen, it was very rare in the early atmosphere, at least in the free-floating gaseous form that composes 21% of the atmosphere today. Volcanoes were commonplace, erupting at up to 100 times the rate they do now. There was so much carbon dioxide in the atmosphere that the surface pressure was 27 times what it is today. This is the equivalent of being 270 meters under the surface of the ocean, where the pressure is it's crushing in its immensity. This carbon dioxide was slowly absorbed into the newly formed oceans, and sucked into the mantle through the actions of plate tectonics. The oceans themselves were boiling pools rich with mineral sediment and the chemical building blocks of life. In time, the surface of this hellish world cooled down. The oceans cooled as well, as undersea volcanoes and magma exposing breaches in the ocean floor shrank, cooled, and solidified. The same happened on the drier surface of the planet, wherein volcanic activity had largely subsided, as the tectonic plates had more or less stabilized. Around 4 billion years ago, the conditions of the Earth had cooled and stabilized by a significant margin, and the Hadean Eon came to an end. This gave way to the Archean Eon, which lasted for nearly 1.5 billion years, from about 4 to 2.8 billion years ago. The Archean Eon saw a significant cooling of the Earth from the Hadean. Make no mistake, the Archean was much hotter than today. There was still significant heat left over from the early bombardment, and the tectonic plates were still in the process of cooling. During the Archean, the tectonic plates cooled enough to allow stable continents to form. There's a general lack of minerals that have been recovered and dated to have originated from the Archean Eon, as the crust is largely believed to have still been relatively tumultuous and susceptible to the occasional resurfacing. After the moon formed, both the moon and Earth happened to intercept a massive cloud of debris. This interception is called the Late Heavy Bombardment, as the young moon and the Earth were engulfed in storm after storm of asteroid impacts. These impacts were really frequent. Based on the dating of crater clusters, it's believed that the Late Heavy Bombardment impacts lasted for nearly 300 million years, from 3.8 to 4.1 million years ago. You thought a 3-hour lecture was bad, or getting stuck in traffic for your whole afternoon. Try getting absolutely devastated by asteroids for 300 million years. The cause of the debris cloud that caused this late heavy bombardment is unknown. It could have been generated by perturbations in the orbital resonance of the outer planets, pushing material into the inner solar system. It could have been formed from a smaller rocky planet, either crumbling from the stresses of gravity or a collision with Mars. What's much better understood is how this late heavy bombardment affected Earth. It's believed that this bombardment was so violent that it contributed to a remelting of some portions of the Earth's crust. During the early Archean, the Sun was 4 billion years younger. At this point in its youth, the Sun was smaller, with about 75% of its current luminosity and heat. This younger Sun, however, put out significantly more ultraviolet radiation than it does today. It isn't quite understood if this would have helped or hindered the development of life. On one hand, the UV radiation could activate some biomolecules and would encourage mutations in the DNA of the earliest cells, promoting genetic variety and encouraging evolution. But on the other hand, depending on its strength, the UV radiation could destabilize or outright destroy most biomolecules. Lightning is thought to have been a much more likely trigger of biomolecule synthesis and activation. This is because lightning can provide enough energy to activate many biomolecules, and can chemically react with the atmosphere and bodies of water in ways favorable to the formation of biomolecules. Liquid water existed on this early Earth, as geologic evidence like pillow basalts and banded iron formations suggest the existence of extremely deep basins. These so-called extremely deep basins are basically what we know to be oceans. It's around this time, around the late Hadean and early Archean, that life is believed to have first appeared. These first life forms were single-celled organisms, primitive cells with primitive molecular machinery inside their barren plasma membranes. The chemical processes through which organic molecules coalesced into working metabolic systems, all encapsulated within the naturally formed phospholipid membranes, it's not well understood. This process, where non-living matter came together to form living matter, is called biopoiesis, or abiogenesis. In layman's terms, it's the genesis of the first life form, of life from non-living matter. These early cells, the first ancestors of all living things, began to multiply. Colonies of cells began to aggregate and move with the tides, with the currents of the water around them, and they eventually got washed up on the coasts of islands and continents. In shallow waters, these microbial organisms, these single cells, would accumulate into groups and form colonies. It's possible that the first cells could have formed in hydrothermal vents, where the steady flow of heat and sulfur would have provided both the thermal and chemical energy to enable the formation of living systems. The minerals on the internal surface of the hydrothermal vents would have provided an ideal substrate, but some people dispute the idea of cells forming in hydrothermal vents, suggesting that UV radiation is a necessary component of early biochemistry. Another theory for the origin of life on Earth is called panspermia. The theory of panspermia argues that the first life on Earth was effectively seeded onto the planet by cells or biomolecular systems delivered here through asteroids. The idea is that microbial life on some other planet or moon or something persisted on a clump of rock as it was blasted out of its planet's gravity well in some kind of extreme impact. In this way, the theory of panspermia doesn't necessarily explain the origin of life itself, just how life came to exist on the planet Earth. This theory might seem outlandish at first, no pun intended, but there's a lot of really interesting evidence that validates the theory. First of all, many biomolecules have been shown to be able to persist for great lengths of time on the surface of asteroids. Second, microorganisms that have been exposed to the vacuum and radiation of space for prolonged periods of time can awaken from dormancy and reproduce like normal. It's entirely possible that life forms like single cells or extremely small microorganisms could survive the impact and subsequent ejection caused by an asteroid impact. Chunks of rock thrown into space in such impacts could be host to these microscopic organisms, and should that rock ever land on a more habitable planet, these organisms could fall off or crawl off and effectively seed the planet. Some people theorize that life on Earth originated from life transferred through panspermia from Mars. It's believed that a young Mars was warmer and wetter, and it's possible that life could have existed at one point on such a young Mars. However, Mars's relatively small size caused its core to cool and solidify relatively quickly. This solidification of the core dissolved the planet's magnetic field, and it exposed the Martian atmosphere to the solar winds. Over millions of years, the Martian atmosphere was degraded and blown away. The surface of Mars was blasted with more and more UV radiation as the density and thickness of the atmosphere shrank. The window of opportunity, so to speak, wouldn't have allowed any possible life to evolve much beyond colonies of single-celled organisms. The planet simply wasn't hospitable to life for a long enough period of time for anything complex to have evolved. It's difficult to precisely identify the first life form and how it originated, but we've identified that life existed as early as 3.7 billion years ago in the form of biogenic graphite found in Greenland, or 3.5 billion years ago in the fossils of microbial mats found in Australia, or stromatolites, which are some of the earliest microbial colonies to live on land, beyond the grasp of the sea. It's believed that the first cells were the ancestors to bacteria. Evolutionary biologists at the Heinrich Heine University in Dusseldorf, Germany, published a study in July 2016 that looked at the genes that were likely to have been present in the LUCA, the last universal common ancestor of all life. This was done in an attempt to see what the LUCA was like, and maybe give clues as to what kind of environment it probably lived in. Their study began with the near 6 million known genes in bacteria and archaea that code for proteins. Similar genes in separate but closely related species are likely to be gene relatives. By organizing these 6 million gene sequences into gene families, the evolutionary biologists did some calculations and were able to narrow down and identify 355 genes that were likely to exist in the LUCA. These genes encoded proteins that suggested a hydrogen-dependent organism that could endure high temperatures, seemingly perfectly fitted for the environment of the inner surface of a deep-sea hydrothermal vent. These genes also included cofactor proteins that used transition metals and ferredoxins, as well as proteins for the metabolism of molecular nitrogen and carbon dioxide, with a dependence on hydrogen. All of these genes suggest an anaerobic organism that lived in a deep-sea hydrothermal vent, where it had ample exposure to molecular hydrogen, carbon dioxide, and iron in the mineral substrate. It should be understood that these 355 genes are not representative of the entire genome of the Luca. It's entirely possible, and most likely, that the Luca's genome had many more protein-coding genes. It's also possible that the so-called Luca detected in the study, the organism which would have likely possessed these 355 genes, is simply one of several ancestors to the bacterial or archaeal lineages. This may have been one of the first life forms, but it isn't necessarily the ancestor of all life forms. The evidence suggests that this particular Luca identified in the study is a methanogenic bacteria from the genus Clostridia. Around 3.5 billion years ago, the first single-celled life forms evolved a biochemical pathway that allowed them to harness the energy of the sun, most notably a group called Cyanobacteria. This biochemical pathway is the original photosynthesis, and it revolutionized life on Earth. Not only that, it revolutionized the Earth itself. You see, this photosynthetic process consumes carbon dioxide, and it produces molecular oxygen. This evolutionary gift is responsible for the current concentration of molecular oxygen in the atmosphere today. The cumulative photosynthetic respiration of all the world's cyanobacteria, lasting for literally billions of years into the modern day, has created the free-floating molecular oxygen that we use to breathe. Of course, all land plants that use photosynthesis also produce this molecular oxygen, but the vast bulk of it is produced by ocean-dwelling cyanobacteria. In the late Archean, photosynthetic cyanobacteria became increasingly common, growing in several forms in the shallow coastal waters on the edges of dry land masses. These cyanobacteria would take forms like mats, where layers and layers of individual cells created a working community. To us, these microbial mats can appear as the slimes that accumulate on rocks sitting on the edge of a lake or a river. Another form that cyanobacteria can take is called stromatolites. The stromatolite is a limestone deposit created in part by the sediment accumulated by the sticky, clumping masses of cyanobacteria. As the sediment builds and the limestone deposit grows, the clumpy mass of cyanobacteria is raised off the ground. These deposits are common in tide pools and other bodies of shallow water. For nearly the entirety of the Archean Eon, for nearly the entire 1.5 billion years, life was limited to this primitive format of single-celled colonies of cyanobacteria. For nearly the entirety of the Archean Eon, for nearly the entire 1.5 billion years, life was limited to this primitive format of single-celled colonies of cyanobacteria. This proliferation of photosynthesizing organisms rapidly leads to the mass production of molecular oxygen, in what is known as the Great Oxygenation Event. Other names for it are more negative, trying to capture the terrible and deadly nature of the oxygen catastrophe, or the oxygen crisis. It's even been called the oxygen Holocaust. You see, the presence of oxygen in the atmosphere was poisonous, deadly in fact, to the numerous species of anaerobic cyanobacteria that lived on dry land and in the oceans. This molecular oxygen reacted with numerous substrates, like decaying biological material and iron deposits exposed on the surface. Notable evidence for these oxygenated iron deposits can be found in Minnesota and Western Australia. When these oxygen sinks became saturated, the molecular oxygen began to react with methane, creating carbon dioxide and water. The concentration of methane in the atmosphere dropped, while the concentrations of oxygen, carbon dioxide, and water vapor rose. As methane is a powerful greenhouse gas, its rapid depletion led to the first ice age, the Huronian Ice Age, which lasted for about 300 million years, ending 2.1 billion years ago. So far in the Precambrian, we've had two distinct 300 million year long periods. One, a fiery hell of asteroid bombardments and volcanoes, and the other, a frozen wasteland of an ice age. This Huronian Ice Age was the beginning of the Proterozoic Eon, the eon following the Archean, which itself lasted for another 2 billion years. It began about 2.5 billion years ago, and it ended about 542 million years ago. Near the end of the Archean, mitochondria evolved. These little guys would go on to play a major role in eukaryotic evolution. The Proterozoic Eon had heavy tectonic activity, just like the Archean and Hadean eons that preceded it. The relative lack of minerals from the Proterozoic suggests heavy recycling of the surface minerals through subduction. Less than half of the crust is believed to have been formed during the Archean Eon, and another less than half-size mass is believed to have formed during the Proterozoic, while less than 20% of the crust solidified in the eon that followed. Life in the Proterozoic evolved in an exponential manner, slowly at first, but accelerating with time. In 2 billion years' time, the single-celled organisms that emerged in the Archean had made numerous evolutionary steps, but 2 of these steps were really big, really important. 2 major evolutionary adaptations in 2 billion years doesn't seem like a very good ratio. But these 2 evolutionary milestones enabled the exponential acceleration of evolutionary complexity among living things. I'll discuss the 2 major steps in a moment, but for now I want to talk a little bit about the mutations that emerged in Proterozoic life. Some of the first cells evolved organelles, becoming the first population in this branching genetic lineage that would become the eukaryotes. The eukaryotes are both larger and more complex than prokaryotes, and thus require significantly more energy. The eukaryotic lineage would go on to make several of its most fundamental branches in the Proterozoic, including plants, which emerged around 1.5 billion years ago, and fungi, which emerged about 1.43 billion years ago. The first cells were all asexual, but when cells began to spread through space and their genomes began to diversify, eukaryotic cells evolved the ability to engage in sexual reproduction. Sexual reproduction offers a number of benefits over asexual reproduction, as well as some drawbacks. Asexual reproduction produces nothing but clones. There isn't any genetic variation, except for the rare and subtle mutation that might afflict every 10th or 100th or 1000th daughter cell. If a disease were to afflict the population, their lack of genetic diversity would mean that if one individual was susceptible, every individual is susceptible. Whole populations of asexual species can be rapidly wiped out by a single disease, or really any other evolutionary pressure that the population can't muster a sufficient response to. Sexual reproduction introduces a degree of variability in the offspring, creating phenotypic variability that allows individuals within a population to have varying levels of fitness in their environments. Populations of sexually reproducing individuals are thus able to produce the genetic variety necessary to survive a disease or adapt to some other evolutionary pressure. Some of the groups that emerged during this period include the Mesomycetozoa and the Choanoflagellates. The Mesomycetozoa belong to a taxon called the Opisthoconta, which overlaps with both the animal and fungal kingdoms. The Mesomycetozoa are small multicellular organisms, composed of just a small handful of cells. These act as parasites for fish and other water-dwelling organisms. The Choanoflagellates are single-celled eukaryotic organisms considered to be the closest relatives to animals that aren't actually animals. The Choanoflagellates possess a small, spherical or oblong cell membrane about 3 to 10 micrometers in length, and a powerful flagellum that it uses to swim through the water. The Proterozoic Eon also saw the diversification of Cyanobacteria stromatolites, which was characterized by variation in the concentrations and types of sediments that they accumulated. Only 600 million years ago, nearing the extremely late end of the Precambrian period, eukaryotic multicellular organisms began to evolve a branching lineage that would become animals. The final 100 million years of the Precambrian Eon were packed with evolutionary change and the emergence of new species. The sponges are perhaps some of the earliest and most simple animals, emerging about 560 million years ago. But other animals, those with nervous systems and muscular tissue that can move around freely, they emerged later. Some of the first complex animals to evolve were the ancestors to the cnidarians, or the modern-day jellyfish and sea anemones. In the last 40 million years before the Cambrian explosion, a group of flat, blob-like animals called Edecaris emerged and thrived. These would become the ancestors of the modern-day arthropods and mollusks. Recall the 2 major evolutionary steps that I mentioned a few minutes ago. The first major evolutionary step would be the development of multicellularity. The earliest known multicellular life comes to us in the form of a Francevillian fossil found in a black shale deposit in Africa. These fossils depict multicellular organisms with radial symmetry and the form of flat oblong discs surrounding a lumpy central structure. Some of these multicellular organisms were up to 12 centimeters wide, which is impressive for creatures that lacked sophisticated vasculature. For comparison, consider modern lichens. These are organisms without vascular structures, and they struggle to grow larger than 4 or 5 centimeters. Other early multicellular organisms took the form of sponges, green and brown algae, and slime molds. The second major evolutionary step would be the emergence of a symbiotic relationship between mitochondria and chloroplasts and their respective eukaryotic hosts. These symbiotic relationships were beneficial to all the parties involved. The eukaryotes that became host to mitochondria took advantage of the ATP produced by their oxidative phosphorylation. The eukaryotes that became host to chloroplasts took advantage of the sugars that the chloroplast could produce through photosynthesis. Both the mitochondria and the chloroplast received benefits from the abundance of nutrients and the inherent safety inside the much larger eukaryotic cell. Both of these evolutionary steps set the groundwork for the proliferation of multicellular life and the Cambrian explosion, which marked the end of the Precambrian, and also the end of this episode. I know I covered a lot, something on the order of the 4 billion years of grindingly slow evolutionary history, but I hope you learned something cool. In the next episode, I'll be discussing the Cambrian explosion and the proliferation and evolution of new physiologies. As always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.